Guys, welcome to my second concept video that I'm posting. I'm actually going back a little bit to discuss the extremophiles, which we talked about on the 7th and 8th as our first discussion section. But I really wanted to just give you guys a resource like that you can refer to later. So if you're studying for exams or reviewing the worksheet or whatever, I wanted to have a recorded version for you to look back at. So going over these slides, which answer the worksheet questions, I'm going to try to do a little bit broader of a discussion. So even though these are the answers to the worksheets, I want to kind of focus on concepts that I've seen people struggle with, or just kind of main themes that I've been getting questions on. So the first question was about thermophiles. So if you remember, thermophiles are organisms that are able to withstand extreme temperatures. It can be, and this definition kind of restricts that to extremely high temperatures. We can also have organisms that are able to withstand extremely cold temperatures. So we have like organisms that can live like under really thick layers of ice in Antarctica, and essentially like are able to like absorb nutrients like just from the air rather than like needing sunlight or anything like that. So it's pretty cool, the things that these cool organisms can survive. Another thing, actually, I want to show you guys this picture. I don't know if you guys know what this is. This is a tardigrade, sometimes called water bears. But they are like bears. They kind of look like bears, but they also can hibernate and remain dormant for really long periods of time. These things are like really hardy suckers. Like they can survive lots of different environmental constraints. They can survive a whole range of temperatures. They can survive without food or nutrients for long periods of time. Lack of water, like dehydration, like basically any stress that you could throw at it, it's going to be able to survive for way longer than almost any organism that we know of. So yeah, that's why there's a picture of it. Okay, and another point that I just wanted to hammer home again, I know that you guys are probably getting annoyed of hearing me say this, but really just trying to think about the relationship between structure and function in these different organisms, or just in general. So it's not enough to say, okay, we know that adding more hydrogen bonds confers resistance to high temps. It's true, and it's a fact that you should know, but we need to go a little bit deeper and explain, okay, so what about those hydrogen bonds is allowing that resistance to those higher temperatures from even beginning? So this is a picture of a protein. So this is its tertiary structure. And we can see that there's different types of bonding that can happen that will allow the tertiary structure to change. So we can see there's hydrogen bonds over here, there's van der Waals interactions here. So this is the hydrophobic region, disulfide bonds, ionic bonds. Not all proteins are going to have all of these that are affecting the tertiary structure, but it's definitely possible. So this is like a kind of a wide range of the possibilities. But we can see how the structure affects function in this case, because having these hydrogen bonds being formed causes that whole segment of the protein to like lift up and create this extra little pocket here. If we were to cleave these two bonds, this whole segment is going to fall down and we're going to lose that shape. And the reason why that's important is because if you watched my enzyme kinetics video, which hopefully you did, we know that so much stuff is related to shape. So if we require, or if we had an active site, if this is an enzyme and there's an active site that somehow, we don't really need to know why, or we don't really need to know the specifics because we're just using an example enzyme, and it's going to be very unique to each enzyme about what's specifically happening. But like say that there was an active site in this little pocket that required something in that pocket, which was what allowed the enzyme reaction to occur. If we get these bonds cleaved and we lose that shape, then we're no longer going to be able to take advantage of that function, which is ultimately what's going to cause like cell death. So if we have we have these high temperatures, which will cause these bonds to cleave, which will cause the shape to change, which will cause the function to change. And if the function is very important, then eventually that will cause the organism to die. So that's kind of like the larger chain that I want you guys to think of. So instead of thinking, okay, more bonds equals more resistance, I want you guys to think of, okay, what are the steps in that? And does that make sense for you conceptually? So hopefully it's becoming a little bit more clear about why that's important. So another issue with bonding that relates to temperature resistance is the hydrogen bonds that form between guanine and cytosine in the DNA of the cell. So we can see that we have an extra hydrogen bond here between the guanine and cytosine compared to the adenosine and thymine, or adenosine and uracil if we're talking about RNA. And so initially when scientists were first discovering extremophiles, because relatively recently, we discovered that they even exist. Previously, we had never thought that there was going to be life at, the crest of these like hydrothermal vents and sulfur deposits and all these things. Like, we didn't realize that. So one of the first hypotheses was maybe there's a systematic difference between extremophiles and non-extremophiles, where these organisms that can withstand high temps, maybe they have significantly more GC base pairs, like as a whole, compared to non-extremophiles. This turned out to not really be true. I keep saying we, not me personally, but science in general has found out that like extremophiles as a whole don't necessarily have a higher GC content in their DNA sequences than non-extremophiles. But that's okay, because we know that there are other mechanisms that allow for temperature resistance, like the ones that we just talked about here. So it could just be that there are other things that are offsetting the GC, like if they have a relatively low GC content, they could still have other structural advantages that will allow them to be extremophiles. Another big thing that affects temperature resistance is fatty acid chains. So we can see here we have this saturated fatty acid chain. So you can see that there are hydrogens on both sides of all of these carbons, or on all the sides of all these carbons. Whereas here, instead of having two hydrogens coming out, we have this extra double bond. If you've taken chemistry, I'm not going to go into the details about why this double bond causes a kink, but this causes a kink here. So again, this is an example of how a structure determines function. So we have this bond, which changes the shape, which then changes its function. How? You can see here. So this is a phospholipid tail that is what is used to create membranes. So we can see, okay, if we have a phospholipid tail that has saturated fatty acids, we're going to have that straight, narrow tail, as opposed to this one that kind of looks like a figure, like sticking his leg out. So we can imagine when we're making membranes out of this extra space that this takes up is going to cause functional changes in how the membrane works. So we're going to have membranes that are composed like this. Having fully saturated phospholipid tails is going to make the membrane relatively like tight, like pushed together. It's going to be less permeable to outside like solutes or whatever. And it's also going to be able to like withstand higher temperatures just because it does have that extra like bit of strength. So when we have this unsaturated layer, we're going to have more space in the membrane to kind of like, they're kind of pushing each other away a little bit. So that's going to make the membrane more permeable, but it's also going to make it like more flexible and fluid, even if we have these really cold temperatures. So if you're talking about like an extremophile that's living in the Arctic or something, if you have this super tight membrane that's not able to pick up nutrients very well, and it's not super fluid, it's not going to survive in that environment as well as one that has a lot of this unsaturated content. So this is just important because I want you guys to remember that like extremophiles can work on both extremes, right? So I don't want you to think, okay, fully saturated is better because you'll be able to withstand higher temperatures. It's true, but there also is an advantage for the unsaturated fatty acids because there are environments which like either or is necessary. Okay, question 2 is about acidophiles, so acid-loving organisms. I think that's kind of a misnomer. I'll explain why, but essentially, if we think about how acidophiles work in real life, it's not that they're able to somehow mitigate like the damage-causing effects of acids inside of their cells. Instead, they operate on a more defensive approach. So they are just protecting like the outer layer of the cell from even getting the acid to enter to begin with. Or if the acids, the extra protons are able to diffuse through the membrane, they immediately are kicked back out again. So essentially, the acidophiles are always going to maintain this neutral pH in their cytosol. So they're never even going to get to the point where the acids can cause damage because they're so much on the defensive. So I really wouldn't call it acidophile because they don't love acids; they just want them out. But they're able to protect against them really effectively, which is why they're able to withstand these environments. So the way that they do this, there's two main ways. The main way, or one of the main ways, is a proton pump. So here we can see an example of a cell where here's the outside solution. It's highly acidic. We have all these protons floating about. And then we have this intracellular environment. And ideally, it's like neutral right now. But say a proton gets through this membrane. And so now the cell is going to be like, oh, shoot, we have a proton, an acidic proton. So what's going to happen is it's going to use this proton pump to push across the gradient. And we can see that there's a gradient because there's a lot of protons out here and there's none in here. So this is why it's going to require this ATP input. It's going to, because it's working against the gradient. And it's going to push the proton right back out again. And this happens really quickly. So this is going to be like right when it comes in, it's going to just be sent right back out again before it can cause any damage. Now another way, another defense mechanism that's even like more of an advanced or like a more of a defensive maneuver, I would say, is having these R groups on the proteins that are like located on the membrane. So if we look at this slide here, this is a good example. So this is a transmembrane protein that we can see, it like goes through the membrane, but it has an extracellular portion and an intracellular portion. Now the extracellular portion is going to kind of act as like a magnet for all of these extra protons floating about. So we can see, this picture is really good. We can see that a lot of these R groups are going to have this extra spot for a proton so that it can become a protonated form. So we're going to be moving between these two forms. If we were on the intracellular side, which doesn't really have protons in there, you'd mostly be in this deprotonated form, which is what we would see on this side of the cell. But when we're outside the cell, when we're looking at this part of the membrane, this top side, we're going to be attracting a lot of these protons that are existing in the solution. So it's going to be a lot in their protonated form. And the reason why this is helpful as a defense mechanism is that it essentially just distracts the acids from going inside the membrane because they're going to be really strongly attached to these R groups instead. So that's a way of preventing them from ever even entering. So it's like one step ahead kind of the proton pump, because the proton pump is just like a really quick like prevention of damage, but this is preventing them from even entering. I think that's all that I wanted to say about that. So I'm going to move on to halophiles. So halophiles are salt-loving organisms. So if we go to the Great Salt Lake or the Dead Sea or something like that, we would still see organisms there. So before I go into this question, I wanted to talk a little bit about what damage salt causes in general. So if we're talking about things that aren't halophiles but are just regular organisms, why is having like salts in the environment bad? And the reason why I want to talk about this is because it's not, it's really different than from like the acids, the acidophiles. So the actual presence of salt itself is not the issue here. Salt doesn't cause damage directly to the cell. Instead, what's happening is, I'll show you these pictures. It's actually due to its dehydration effects. So if we look at this picture here, we have this hypertonic solution. So the inner part of the cell, the intracellular component, has a relatively low concentration of salt compared to the outside solution. And obviously with diffusion and osmosis and stuff, we want to maintain a equilibrium. So we really don't like having this difference between solute concentrations. An important point to note for this is that we have this semi-permeable membrane here, which restricts what we can do to solve this concentration difference. So if you think about, okay, what if we had, instead of a cell, what if we had just like another beaker of water? If we had another beaker of water with a relatively low solute concentration, and this was in a separate beaker with the high concentration, and you mix those two beakers together, what will happen is both the solute and the solvent, so the water and the salt particles, will be able to move around and establish that equilibrium on its own. So eventually, without us even having to do anything, we would be able to test the concentration at different areas of the beaker, and it'd be like relatively consistent throughout. However, we're not dealing with beakers, we're dealing with cells, and so we have this extra membrane involved. And so with osmosis, only the solvent particles are able to diffuse across this membrane. Because you could think like, oh, well, to avoid dehydration, instead of bringing water out, why don't we just bring extra salt in? We can't do that because of the limits of osmosis. So we have to bring water out to neutralize this. And it's going to be just like a natural occurrence that the water is going to go out of the membrane. So it's actually the dehydrating effects that are what's causing the damage. What's causing the damage to the cells. It's not a direct effect like with acids. There also can be damage with a hypotonic solution. It's going to be less common than the effects of a hypertonic solution, but you can have a situation where there's so much salt on the inside compared to the outside that you're getting a major influx of water, and then your cells will burst, they'll lyse, and then they'll die. So you guys might have heard that like they had to ban water drinking contests for like radio stations and stuff because people were dying of overhydration. And that's what was happening, is that you're getting cell death because there's so much water being influxed. But as you can imagine, we're much more likely to die of dehydration than overhydration, but it is still a potential issue. The ideal would be an isotonic solution where we have this nice equilibrium between the inside and the outside of the cell. And just another point to make about this is that we can see that there are arrows here. So that's indicating that it's not like, oh, suddenly we reached equilibrium and now the membrane is like closing itself off and like not going to let anything exchange. That's not what's happening. Obviously the membrane is still semi-permeable to water, but the water is just like flowing back and forth, and like it's moving in rates so consistent with each other that when we measure like over periods of time, it's going to be basically the same concentration between the inside and the outside. So the reason that halophiles are able to survive this is that they're actually able to modify their intracellular solute component on their own. So they're able to sense when they're running low on salt on the inside of their cells and up that solute concentration. So they're going to produce more of these solutes in response to that, which is really cool because it totally negates the effect of dehydration, because they're going to increase the solutes in here until they maintain an isotonic solution, and then they get to keep all their water. So this is a really good idea because because the salts aren't causing direct damage. So if you can just increase your salt production naturally, then you don't have to get rid of any of your water. So yeah, that's how halophiles do that. Question 4. Okay, now we're moving on to something a little bit different. This is more of like a logistical question rather than a molecular question, which I think was confusing to some people. But so like, why is it difficult to culture large quantities of extremophiles, especially like for industrial use? And the reason, it might seem kind of obvious now, but it's literally just because it's too difficult to maintain, or it can be like really difficult and really expensive. If we think about the types of environments in which extremophiles live, we're talking deep sea hydrothermal vents that like we didn't even discover life until like the 70s there. These really deep Arctic sheets, sulfur deposits, all of these things that are very either dangerous or hard to mimic. So like mimicking the pressure and the light, the absence of like light and air and stuff from these like really deep pressurized caves, those are not going to be really easily reproduced. Or even if we try to reproduce the conditions, we might not actually be simulating what's really going on in real life, even though we think that we're like mimicking the pressure conditions and stuff exactly right. So it's just difficult, and it's also, I mean, a lot harder than just trying to grow a culture of E. coli on your desk. Okay, so question 5, how can biologists tailor extremozymes? So why would we want to tailor extremozymes to begin with? Well, we can look back at question 4. Getting the ones from nature that exist can be really challenging and expensive. And more broadly, why we would want to do this to begin with is just because there's so many cool applications that we can use them for. So if we think of Taq polymerase, which is that extremely heat-resistant extremophile, thermophile, when we discovered that and started using that in polymerase chain reactions, we were able to increase the efficiency of PCRs like by many orders of magnitude, and it revolutionized how we conduct biology today. There was like an instant Nobel Prize for like developing that with the use of an extremophile. So that's why it's important. So the two main methods. The first method that I'm going to talk about is rational design. Rational design is kind of where tailoring enzymes started, where it began. We eventually moved on from it because it doesn't really work that well, but I'll explain why. So rational design is essentially picking structures that we know offer certain advantages. So like the ones that we talked about earlier, like adding, like if we wanted something to be more thermophilic, we could think, okay, maybe we want to add more van der Waals interactions and more fully saturated fatty acids. Maybe those are some things that we can do to design this enzyme. And in theory, that sounds great, but in reality, it's really hard for us to compete with the complexity of nature. So there are many instances where we would try to do things that we think would really increase the thermophilic capacity for an enzyme, and it just doesn't. Nature is super complex, and there's a lot of things that we still don't know. Scientists are still unaware of lots of things. So generally, they don't really work as well as we think they will. And nature's, like, extremophiles are generally a lot better at functioning than the ones that we try to create on our own. And this could be for a variety of reasons. There could be effects that like we just can't even, like we haven't even discovered yet. There could be like epigenetic signaling and like all of these things that are kind of blind to the human eye currently that we just don't know about. Or there could just be kind of like the butterfly effect, where these like really, really small things can end up making a huge difference. There could be other things that we're introducing into the mix. So like when we're adding these bonds, when we're adding, like when we're changing different contents of things, like maybe we're also doing something else that's altering the structure in another way, like in the opposite direction, that are just so small on an individual scale that we're just not like noticing. So it's just more complex than it seems. So what we've done to kind of get better at making extreme enzymes, we switched to a different approach. This approach is called directed evolution. Directed evolution is essentially when you are growing a culture and you're growing it over many, many generation times. So if you think about generation times of different like cells, they're generally really fast. Like if we're generating a cell culture of E. coli, you can get 100 generations in 3 days, which if you were trying to mimic that in a higher level organism, that would be extremely hard to do. Like that, you wouldn't be able to see that in a period of like many human lifetimes. But doing that in this case, we're able to get so many generations so quickly, which is helpful because we know that evolution works by just spontaneously generated mutations, and some of them are advantageous in certain conditions. And so what we're doing is we're taking advantage of these generation times to look for any of these mutations that occur naturally, randomly, that can be like effective for what we're looking for. So the way that we do this, the way that we're able to find these cells with these mutations that we want from such a large cohort, like we have all these generations of cells to use, how we can narrow that down is by using this thing called selective media. So selective media is essentially just growing things in the condition that you want to like test against to see what is able to grow. So a common example of this is like antibiotics. So if we have a cell culture that we want to test to see, okay, which cells have developed a resistance to antibiotics, what we would want to do is grow a bunch of those cells in antibiotics, because then any ones that grow are going to be the ones that are resistant. So we know that something about it has changed that allows it to survive. We don't know the mechanism yet. We could sequence it. We could try to do different tests to see what the mechanism is, but like we don't know what it is, but we know that they can grow. So something is different. And so we essentially use that theory to our advantage. We say, okay, we want something that will be able to grow in higher environments. So we're going to culture all of these cells in high environments. Anything that can grow are the ones that are going to have the properties that we want. So the advantage of this is that we know that it works. We've already seen an example of it growing in real time, like in these conditions, which is really important, especially when we're talking about like medicine and biotech and like things where these actually like make huge differences in human lives. Like we don't really care that we don't know exactly what caused the mutation. We just know, or like what the mutation actually is or how it's affecting it. We just know that it works. So that's all that we care about. It does come with the disadvantage of directed evolution is that it does come with that like relinquishing of control. Like we don't know what's in it. We don't know what happened. We can try our best to look at the sequence and try to deduce what's going on, but we don't know for sure because we didn't engineer it from scratch. So we do have that like lack of clarity a little bit, but I think it comes at a really fair cost of being able to find extremozymes that actually work. So hopefully that makes sense. Question 6 is really similar. I basically just talked about it, but it's just saying like, why can't we make our own extremozymes from normal enzymes? And that's just because, like I said, the structures are like, there's so many different little tiny effects, these like butterfly effects that we just don't have full control over yet. Like we can tell when we're comparing an extremophile versus a regular enzyme in real life, we can say, oh, these are like the major differences between the two. But there's also so many other factors because there could be other non-extremophiles that are super similar in structure to them that we just haven't been comparing yet. So we can't see the difference between like those really, really similar ones to see what's causing that difference. So like we're just kind of confined by what we can study and what we can grow. And yeah, so that's why it's a little bit of a struggle, but I think over time we are going to improve with developing our own extremozymes. So it is something to kind of keep an eye out on if you guys are interested in this topic. I love just hearing about extremophiles. There is some interesting articles about like a new type of bacteria that was found in 2017 that doesn't require anything to live except for air. So like it doesn't need any sunlight, it doesn't need water, like it's just crazy. Like there's all these, it's just like one of the only organisms, I think the only organism that was determined to only need air to survive, which is really cool. So the rest of these slides are kind of just the key concepts of what I've talked about. So I'm not going to go into them in super high detail. But the reason why we put them on here is because when you're studying for the exam, things like that, what would be really helpful for you is to go through these key concepts. And when you look at the like, kind of short bullet point list, if you're able to connect that to like, okay, what did we learn about this? Why was this important? How does this work? If you're able to explain it, if you can like articulate it to yourself out loud, that will tell you, okay, cool, I like totally understand this concept. Whereas if you look at like this slide, for example, and you say DNA, okay, thermostable, proteins, higher GC, and if that doesn't ring like any bells to you, I would go back to the previous discussion and like look more in depth, and that will tell you like where to focus your studies on. So I hope that helps. But essentially this just goes through all the questions again. Yeah, I think that's all that I wanted to say. So anyway, thank you for watching this video. I hope that it was helpful for you. Again, if there's any feedback that you guys have for me, please don't hesitate to reach out. Yeah, we're gonna have another discussion on Gibbs free energy. So I'll be posting that video hopefully by today, but that's our next topic for discussion. So, thanks guys.